Hello and welcome to GeneLab Chats. Today we'll be interviewing Dr. Giacomello from SciLife Lab and KTH Royal Institute of Technology in Stockholm, Sweden. It's well documented that the space environment adversely affects biological systems, and molecular methods have been instrumental in providing more mechanistic information on this phenomenon. Currently, the typical method of spaceflight omics analysis involves whole tissue processing, which provides much of the known information on tissue-level understanding. Single-cell RNA-seq and spatially resolved transcript omics are examples of higher resolution methods that can provide more information about constituent cell types. But both of these methods have not yet been optimized nor standardized for spaceflight samples. In this interview, Dr. Giacomello presents research and highlights how methods and techniques discussed in this publication can enable future GeneLab data generation. Welcome, Dr. Giacomello.
Thank you very much.
Let me start off by asking you to please let the audience know what were the most important recommendations in your study.
Yeah, so in our manuscript, so we have presented how we should collect and preserve spaceflight samples in order to perform single-cell RNA sequencing and spatial transcriptomics type of analysis. So that is for sure one of the main aspects that we touch upon in our article. So for example, in order to collect properly samples, we really need to be careful on how dissections are performed on mice, and also how these organs, once dissected, are handled in order to preserve the morphology of the organs if we want to do spatially resolved type of transcriptomics analysis, where indeed the morphology is key, or also how we preserve samples if we want to do single-cell RNA sequencing analysis. But in both cases, it's essential to have great quality of the RNA plus the morphological aspect for spatial type of analysis. And another aspect very important in our study was related to our suggestion on how to conduct spatially resolved transcriptomic studies, because they are expensive from one hand, and on the other hand, spaceflight samples are rare and unique. So you don't really want to waste the material that you have in your hands. So we really recommend future scientists that want to perform this type of analysis on such samples to have a testing cycle in which there is first an evaluation of the type of tissue that we want to study. So for example, which specific regions of the different organs needs to be analyzed, and how to conduct the experiment, as well as performing and test experiments in order to make sure that everything can run smoothly before moving to the real spaceflight samples.
That's wonderful to hear, and thank you. And my second question for you is, how will the GeneLab database assist you in your upcoming work?
Yeah, so I must say that this work, or more than the work, the whole knowledge that we have then presented in this perspective article of in cell report methods would have not been possible to acquire if we didn't have started the collaboration with actually NASA GeneLabs, where we have started to look into mouse brains and hearts, because just really by doing the work, we were able to understand how in previous missions, and therefore like legacy spaceflight samples, the morphology has not been preserved as accurately as it should be, or the preservation also of the RNA, how that was done, and what type of new fixatives, for example, can be utilized. So I must say that, yeah, the collaboration, it has been really instrumental in order to learn all these aspects in order to implement future experiments on spaceflight samples.
Thank you again for that, Dr. Giacomello. This was very informative, and I look forward to your upcoming work. To our audience out there, you can learn more about this work, how to join the analysis working groups, and access all the data sets presented in this work with the links provided in the description section below. Thank you and have a wonderful day.